Hi, everyone. I'm so sorry for this delay. It turns out that there were two Zoom links floating around. And so everyone on the leadership team that's going to be presenting was on one link, and then all of you were on a different link. So I really apologize for the delay. We're going to get started as soon as I have everybody admitted into the meeting. So again, apologies for that technical difficulty. We had two different links floating around. I'm so sorry about that. See, leadership team, this is what I was expecting to see in terms of participants, not one person. We knew there was something wrong. Okay,
I
so
think it's a
we're,
bit, I don't know if sorry, just to
yeah,
say we're all
please.
called Cassandra Brooks on my screen.
Oh,
That's so strange.
I am
But
seeing a lot of
Probably because
Cassandra Brooks.
probably because I forwarded the
Well,
link to all of
Cassandra
you.
too.
But you should. This is super fun. Actually, I love this. Just keep it. No, if you go to the you can change your name. Or you can
Sorry,
keep it, you know.
this is the strangest Zoom experience I've had in a long time. So, thank you, everyone. Thank you, Cassandras, for your patience. Um, I'm going to get started right away, just to introduce the meeting and get us started. And again, apologies for that delay. My name is Jill DeCacavo, and along with Cassandra Brooks, we co-lead SCARFISH, the action group on Antarctic fish within SCAR. I believe many of you are already familiar with the group. Just to give you an overview of what we're going to be talking about today, I will briefly, 2 minutes, introduce SCARFISH to all of you just to make sure we're on the same page. Then we'll have representatives of each of our 7 working groups present around 5 minutes on what they're doing, what sort of outputs they're working on, and where there is possibility to get involved in those groups. Those working group presentations will be followed by Cassandra presenting on CAMLAR, very briefly bringing up everyone up to speed on what CAMLAR is, but mostly talking about what SCARFISH is doing with CAMLAR and the sort of latest big issues and actions coming out of CAMLAR in 2025. All of that will be followed by a question and answer session. So hold on to any questions or comments that you might have regarding both the working group presentations as well as the CAMLAR presentation. And we'll have time at the end of the meeting to have more of a question and answer and discussion. So with that, I will share my screen and just briefly introduce SCARFISH. Okay, so hopefully you're seeing my screen. Um, Continue to share. Okay, hopefully you're seeing my screen there.
We're seeing your presenter view
Presenter
though,
view. Okay, what
Shilda.
if I do that?
Yep, that's great.
Perfect. Okay. I'll just look at my other screen. So perfect. So as I mentioned, we're SCARFISH. The list here also includes the members of our executive group. Um, and so what our motivation, of course, is the importance of fish, as I don't think I need to tell any of you, in the Southern Ocean, and the fact that there didn't exist a group dedicated to fish in SCAR. And in many ways, we took inspiration from the SCAR action group that was created on krill. And that is now an expert group. But we sort of wanted to our scope is even larger because rather than just being focused on one species like skag and skeg, the krill group, we want to have a focus on all Antarctic fish and not only just those that are exploited, with not only a goal to bring together this diverse research community um, in ways to help facilitate research and address gaps in Antarctic fish science, but also to help to inform policy, working with one of our biggest partners, of course, which is CAMLAR, the Commission for the Conservation of Antarctic Marine Living Resources. Um, and so, yeah, I think I probably went ahead of myself and mentioned sort of the first two, but also the point on broadening diversity is another important one that we're aiming to do in our group. So it's about bringing more, not only different levels of career, but also representatives from different countries to come to the table, in part because, for example, CAMLAR only has a certain number of member countries that are involved in it. And so our role as SCARFISH is to also bring in research and ideas that are coming from areas perhaps that are not as well represented in CAMLAR just because of its structure. And so far, our group, so the way SCARFISH is functioning is that we have a sort of executive group that manages our different working groups. And so you're going to hear more from the working groups coming up, but the working groups are really the ones that carry out the activities of SCARFISH. And the people involved in these working groups, we try to really go across all different axes of diversity from career stage, even having industry representation, global representation, et cetera. So these are the 7 working groups that we're going to be talking, we'll have representatives talking to today. So I'm not going to go into too much detail because I'll let them speak to what they're doing. But just to give you ideas sort of the number of people signed up to each group, and each group is going to tell you today where there might be space to still get involved at this stage. And so sorry, that's a bit cut off, but this will be a message repeated again and again that you are able to sign up to our listserv, sign up to one of our working groups. All of the working groups are going to share information on how to do that. So without further ado, I'm going to stop my share and hand the floor over to our first presenter, which I believe is Jose.
Yes, Sheila, thank you. I just sent you a request, I think.
Should be good.
Cool. So we'll see now the screen, correct? Yeah, cool. Yeah, so thank you, Gilda, and hello, everyone. So I'm here to present a bit about the work that we are doing on the Horizon Scan Working Group. So together with Gilda, Cassandra, and Anton, we are co-leading this group. And basically what I'm going to show you today is like why we are doing this and the timeline and where you can join the group. So basically, the objective of this working group is to identify the major knowledge gaps and prioritize the main research questions about the Southern Ocean fish. And to do that, we'll do something called an horizon scan. But we all ask sometimes, what is this? We saw it sometimes in titles of articles. But the question is like, this horizon scans is an established mechanism that allows to identify issues, threats, and future directions of a research topic or a discipline or even a region. For example, we can have a horizon scan on cephalopods of the Atlantic, but we can also have a horizon scan, as we'll see later on this presentation, on the research in the Antarctic. It includes, it includes a group of experts and wider community. So the group of experts usually is used for the refinement of the input that we receive from the wider community, as the objective of these horizon scans is to gather the highest possible consensus on a topic, for example, these priorities, and having everyone involved in the process is the best way to have the highest consensus, the widest consensus. And in the end, the horizon scan is useful for planning, to have policy initiatives, but also to define the research priorities. So in terms of the Antarctica and the Southern Ocean, the main horizon scan that was done until today was in 2014, led by Kennicott. I don't know, it's Malone Kennicott. It was in 2014. SCAR is updating it now, so you might have received an email a couple of weeks ago from SCAR to contribute for this. But since this horizon scan in the Antarctic, the general research in the Antarctic, we had different ones targeting different areas of the Southern Ocean, different disciplines, different groups, just focusing on a specific technique. But indeed, and despite the role that fish has in the Southern Ocean as prey, as predator, wherever you name it, this remains overlooked. So the community never come together to have an horizon scan to define the priorities for the Southern Ocean fish. And this is where the SCARFISH Horizon Scan Working Group comes into force. And in terms of what we are going to do, we'll use the Delphi method, which is an established method to do the horizon scans. It basically works with a series of community questionnaires that is followed, as I mentioned before, by a refinement by the expert group. So in our case, what we'll use is like the SCARFISH Horizon Scan working group members as the expert group, and the wider community that we'll use will be mostly the SCARFISH and SCAR lists, but also the group of practitioners interested in the Southern Ocean fish. In this case, we'll send it directly, for example, to Camlor, to Comnap, but then we'll also share it widest possible using, for example, the SCAR social media. So everyone is invited to participate, to contribute on these questionnaires, and then the expert group is here to refine all your thousands answers that you'll give us. We also aim to share it during the SCAR Open Science Conference in Oslo later this year, but this is dependent on the acceptance for the abstract for the poster. So we'll see. Hopefully, we'll be there. So in terms of the timeline, since the beginning of the SCARFISH, what we have done is like looking for previous horizon scans to understand which were the priorities identified in these documents and to prepare a series of questions based on these horizon scans that were published in the past, but also new questions that came from the mind of the expert group, and we draft the first questionnaire. So the idea now is to send your first questionnaire to everyone in the community. This will probably run between March to August 2026. So March is like we'll try to do it as fast as possible. August is so we can actually reach the end of the SCAR Open Science Conference. This questionnaire is based on open questions, just asking people what are the priorities they identify for the Southern Ocean fish. Then we'll have the expert group revising the answers of the first questionnaire and trying to draft the second questionnaire that will be sent hopefully on these dates until February 2027. And in this questionnaire, the idea is to prioritize the priorities that people highlighted. So we don't want like a list of 1,000 questions. We want a short list of priorities so everyone comes together trying to answer the same thing and not continue as the same as everyone has their own question. And then the last two steps is like review the second questionnaire and identify the priorities. And in the end, hopefully by the SCAR Biology 2027, we'll have the horizon scan for this for the Southern Ocean fish. So to finish this presentation, just to highlight that we are now 38 members, 10 of us are early or mid-career stage, which is really good. However, we are always looking for new members, so please come aboard. So we always need people to look for the answers and try to understand and pull them together. So feel free to join. This link just goes to the form that Gilda probably added on her presentation too. And that's it. Thank you very much. And
Thank
I'll
you so
stop
much,
sharing.
Jose. That was perfect. And as I said, every, I think, presenter is going to provide like another link or a QR code to join. So if you didn't catch that, it'll come back. And I think we'll also figure out a way to share slides and things like that. So thank you, Jose. And without further ado, I'll pass on to Becky to present on the data group.
Yes, please. 1 second. You should be able to see my presentation, hopefully. Awesome. Hi, I am Becky Kneinberg, one of the many Cassandra Brooks. So I apologize in advance. My presentation doesn't have a lot of pictures. It has a lot of text, which I try to avoid. However, I wasn't sure I would make this meeting, so I went a little overboard just in case that Gilda had to give this presentation on my behalf. Anywho, so I, together with Anton van der Putte, lead the data working group. So again, a lot of text. It is, I always get this question, why do we have a data group? And when you think about it, there's actually several reasons for this. So the first thing is obviously, when you think about Southern Ocean fish, a lot of the issues stem from data, the lack of it, the inaccessibility of it. We've got all the traditional data limitations for the Southern Ocean, you know, that we have limited winter observations, we have generally very few observations in space and time. It also means that the observations we do have are very precious and they're usually very expensive to collect. Then on top of that, we've got the usual gear protocol changes over time. Things are developing even now. We've got new technologies. So trying to keep everything consistent and comparable is a major challenge. So again, this is nothing new for the Southern Ocean. But you know, it is just worth highlighting that this applies to Southern Ocean fish as well. There's some excellent literature on this for the Southern Ocean more generally. If you haven't tapped into that, there's some fantastic papers I can point your way. But then we've also got additional challenges. So we've got commercial data, which a lot of us know is hidden behind various walls for confidentiality. Then the data generally, not everything is uploaded. We ecologists sometimes have a tendency to just leave things in a spreadsheet for later, or leave things in a desk drawer. That makes it really inaccessible for other people. And then even the data that is uploaded, doesn't always have the correct metadata. It doesn't always have adequate quality control. Some data, was collected in the 80s, is being uploaded now. Nobody really knows what happened to it, and they're just uploading it as is. So there's all of these different challenges that we face in addition to the usual Southern Ocean challenges. So part of the reason we exist as a group is so that we acknowledge this and we document this, but also that we can help change it in the future. So obviously, data accessibility is a huge hurdle for early career researchers, for people who don't have a huge support network from their institute, perhaps, or from their country. So really trying to lower this hurdle is what we're after. And then additionally, within SCARFISH, we have other working groups who are producing outputs, for instance, like the Horizon Scan. So we also want to make sure that the standard we hold others to, we as SCARFISH abide by. So we also advise our own working groups just to make sure that anything they produce or they publish ticks all of those boxes. So one of the major things for our group is we are not trying to reinvent the wheel. There is a SCAR data policy, which very clearly outlines some of the core things that we're trying to hit. I've extracted a couple here. Again, if you haven't ever engaged with this document, it's actually not that long and it is very valuable. So it outlines things like data should be ethically open, it should be reusable, it should be free, it should be fair, which is an acronym basically just trying to make sure that it hits a lot of the accessibility standards, for instance. It should be accompanied by appropriate metadata, which should be obvious, but again, in a lot of cases is not. And crucially, it also goes from data formatting to actually data infrastructure, and it says data should be preserved for the long term. And that often involves actually making sure that there's funding, making sure that there's long-term data stewardship. So it's a very huge concept. But again, it's a document that exists. There's several policies from other institutions as well. So that is the environment we operate within. There's entire groups that are dedicated to Southern Ocean topics. So we're very lucky that Anton is involved in a good number of these. And I myself am involved in SEUS as an ECR rep. So we've got the Standing Committee on Antarctic Data Management. We've got the Southern Ocean Observing System. We've got the Polar Data Discovery and Enhancement Research Working Group. And that's just to name a few. When you start looking, there's data groups all over the place. And then again, there's also existing data standards. We're using things like schema.org, Darwin Core. Again, there are things that exist. We don't want to reinvent the wheel. So for instance, Darwin Core is a bioinformatics standard. Schema.org relates to the way that metadata is formatted, that kind of stuff. So that all exists and we draw on it. So what are we actually trying to do? So like I said, throughout our lifetime, we will continuously provide advice to other SCARFISH working groups for any data they publish. And then during some of the presentation, you'll also see some of the working groups are thinking about updating data, for instance, things that are on FishBase. So again, we'd be involved here. We can always use more hands on deck. So where I've marked opportunities, that is really where, if we had more hands on deck, we would really appreciate it, but also don't feel you have to come for a certain task to join us. You're welcome to join us without joining any of the tasks. We've also got data inventories. So for instance, things like the Horizon Scan or some of the other papers, reports, we're really trying to make it transparent where the data is available for researchers. So again, that is a huge task. So the more hands we have, the better. Then specifically for this year, starting into the next, we are also working to identify some of those big hurdles I talked about and be really specific about where they exist and what might be done about them to increase data accessibility. We're still working out exactly what we're going to do, whether that will be a paper, a report, or something else. Again, there's a huge opportunity here. Another thing we're doing specifically this year is we are supporting the IUCN is trying to update some listings for Southern Ocean fish. A lot of SCARFISH members are involved, so there's a lot of overlap, and we're sort of just acting as the liaison, just making sure that everybody understands what data they're supposed to bring to the workshops and that kind of stuff, just to make sure we as SCARFISH are as prepared as possible. So that's currently ongoing. And then on top of that, we're always looking for opportunities. So if you have a great idea, like a workshop or a hackathon or anything of that kind, please come join us. We are ourselves starting to think of some things. Anton is involved in a lot of workshops that are run by other organizations. So we have a direct link there, and we will start advertising these on the SCARFISH mailing list. So definitely join that to hear about all of those. Additionally, something we've done, for instance, last year is we do attend other discussions. So where a fish perspective, fish data perspective might be useful. So for instance, last year I attended the Polar Data Forum. As part of that, I was part of the discussions for the next International Polar Year. Again, we're just trying to make sure we have a presence. We voice some of our concerns just to make sure that going forward, hopefully we have fewer of these data issues. And we're just trying to make sure that, a diversity of voices, including fish data, are included in these discussions. So if you are part of a group or discussions where you would think, oh, hey, SCARFISH data working group might be good to just include, we're happy to just sit there and listen and be informed, but also happy to input. So please do reach out. This is something that we do. Similarly, if you are on a completely different track and you are a researcher and you have fish data that is not published, that has been in a drawer for a while, you don't know how to publish it, just come talk to us. It's not our primary purpose, but we will do absolutely anything to increase accessibility so we can at least point you in the right direction, point you to the right people. I've put below a very excellent document that not many people are aware of. So it's a brief guide to publishing data for the polar research community. If you just type that into Google, it'll get you straight to, it is just a couple of pages in a PDF. It is very step-by-step. It runs you through a lot of those things. But again, if you need any more help than that, please do reach out. We are very happy to help. So instead of the QR code, I've got the link for you. So please do sign up for the mailing list of SCARFISH in general, but also the data working group membership. So you can also email SCARFISH, or you can even email me directly if you would like. The next working group meeting is on the 3rd of March. So if you add yourself to the mailing list now, I might be able to add you to that meeting, and you can come have your first SCARFISH data meeting with us. Anyway, that is all I have got.
Thank you so much, Becky.
The IUCN.
You
I'm
know,
so good with all my other acronyms.
No worries. I just thought it would be good to throw the acronym in the chat.
Thank you.
Amazing. So just everyone remember to note your questions and comments or anything regarding each of the individual working groups for our discussion at the end. And I will pass over directly to Haley to talk to us about the fieldwork group.
Thanks, Gilda. Let's pull up here. Are you all seeing my slides or presenter view?
Seeing your slides.
Okay, great. Hi, everyone. My name is Haley Kwasniewski, and today I have the pleasure of presenting different work from the Fieldwork Working Group on behalf of myself and my other two working group leads who are also here, and a bunch of other wonderful members in our group that have made a lot of progress, and we're doing a lot of work. So I'm really just so happy and honored to be presenting on behalf of all them and what we've all been up to. And so today I'll just chat briefly about what this fieldwork working group is currently doing and where we hope to go. And so overall, our fieldwork working group has, I would say, 3 main goals. And the first one being increased knowledge sharing among Antarctic fish researchers and within the community. And so improving just sharing information about things like planned field work, potential sample availability, or other collection opportunities to just kind of improve communication among the research community in this way. We also want to emphasize data standardization and actually obtaining data in the field in a certain standardized way, just so that once we all do have data, it's more readily comparable and shareable, which definitely ties into what we just heard from the data working group for sure. And lastly, we hope to develop guidelines for and best approaches to fieldwork practices. And so these can include resources like safety plans and protocols, some sampling guidelines, as well as just other general resources to help make Antarctic fish research and field work a little bit more accessible and kind of decrease some of those barriers and make some knowledge more publicly available. Oops. Sorry. But in order to meet those goals, we've been working on a few things. One being a literature review on Antarctic fish sampling methods. We hope to submit that by this summer or during this summer, which is really exciting. This is kind of the start of the conversation for standardizing those field methods and also just producing something that is public and accessible for people interested in Antarctic field work regarding fish. And then something that I'm really excited about, I think, is this SCAR Open Sciences will be announcing a sample sharing platform. And this was really created to connect Antarctic fish researchers together in the community. This platform will allow folks to either request potential samples from people who've been in the field, or it'll allow researchers who have been in the field and maybe have extra samples kind of lying around to post them publicly and kind of solicit collaboration among the community. And so this is really to kind of get at that, some people, we might all have some samples lying around that aren't being used, but we could really love to be a part of a collaboration to use those and put them out into the world. And so I think this will be a really exciting way for people to connect with each other. And then building from that, we hope to kind of create this toolkit for Antarctic fish field work that includes things like best practices for fish sampling, standardizing sampling methods, and different resources and tools for like field safety plans, and just help people get those resources that make field work a bit more accessible and a little less challenging to enter, I guess. And so overall, we have a lot of people helping us in the working group, but like everyone has said, we could always use more folks involved. And we've got a lot going on, so really anyone can plug in anywhere. And while I don't have a QR code or link, I do have our emails, but I'm sure those QR codes will be circling around and probably linked in the chat at some point. So with that, thanks for listening. And of course, if anyone has any questions during the Q&A portion, any of the three of us will be happy to answer those.
Thank you, Haley. And I will now be presenting on behalf of Manuel for the outreach working group. Manuel is unfortunately on field work this week, and so sent me his slides in advance. Okay, so hopefully you are seeing the actual screen. Perfect. So outreach group. So their goal is sort of multifaceted. They not only want to produce materials with a sort of public engagement object, but also with respect to potentially policymakers, and also about the work that SCARFISH does as well, sort of to improve the transparency of our work and make sure that the community knows what we're doing and that we're not sort of working in the dark per se. They had generally conceived of this as something to be done basically through the SCARFISH website as well as through social media. And what we've been discussing about this is that a lot of SCAR groups actually use the platform that SCAR has as a wider organization to perform these types of communications rather than creating our own SCARFISH website. I mean, we have a SCARFISH website, but that is within the SCAR website, a section of the SCAR website. And similarly, SCAR has a whole social media presence with thousands of followers and things. So we don't necessarily need to create our own. SCAR encourages its groups like SCARFISH to provide them content to share. So the idea is that the outreach group would use SCAR's existing network and existing web platforms to facilitate this type of communication. And so especially in the beginning of the group, there was a lot of discussion about creating university materials. I think this sort of really will depend on the people that are involved in the group and whether they're involved in teaching and have the capacity to provide that sort of effort into doing that. We are trying to also develop a collaboration with Polar Educators International, which would also be in the vein of creating public geared materials to share for outreach. And then finally, with respect to the policy section, I think the ways in which the outreach group was thinking was a bit more like on the side of policy briefs. What is it called? The environmental portal that SCAR has, which is this part that these policy briefs and sort of very shiny, nice infographics and things and articles that try to break down certain concepts or ideas or outputs from a given SCAR group in a way that is really easy to uptake by policymakers. So that was another way that the group was considering that they could create these types of communications in the sort of more policy-focused sphere. So Manuel will be back next month from fieldwork, and so they'll be planning their next meeting then. So if you're interested in getting involved in the outreach and communication part of SCARFISH, I would encourage you when you sign up to working groups to click on the outreach group, and we'll add you to their mailing list, and you'll get all the information for their next meeting. And I will be sure to, once I finish this presentation, put that link again in the chat to the form for joining working groups. And that is a form for all working groups, and you just pick which ones you want to join, and you'll be added to those groups' mailing lists. And I think that was it. Okay, so with that, I will then hand over to Edika.
Yes. Okay. Okay, is okay? Can you see my screen?
Yes, we can see, and it's
Okay,
the good view.
Okay, thank you. Hi, everyone. I am Erika Carlig, and together with William Reed and Dale Machette, I'm co-leader of Biology and Life History Working Group. The aim of this group is to summarize and standardize the key biological and life history traits of southern ocean fish, identify critical habits and periods, develop a traits database, and finally supporting assessment models and ecosystem management. The working group has two priority areas. The first, improving our understanding of the air life history stage. And the second, undertaking a systematic review of life history, including reproduction, recruitment, growth, feeding ecology, etc. Sorry. Before Christmas, the working group led a session focused on planning the systematic review on the life cycle and the biological traits of Antarctic fish. During the meeting, we discuss the information to be collected, the scope of the review, and the next steps. We drafted a Google spreadsheet outlining the key biological traits and the instruction, and starting adding instruction for extracting data for peer review literature. Given the art of the task, we have created a form to get understanding of where people would like to help with the process, and we would be grateful if you could fill out the form by Friday. Finally, we would like to invite you to join in the workshop of the Southern Ocean Species Treats. Will be organized by SCAR and Worms. As leader of this group, we will participate in person. The workshops aims to make species traits data public accessible by providing hands-on training with platform such as Fish Base and Worms. Diciamo, in conclusion, we welcome new members and invite interested colleagues to join us. Please contact us. Thank you so much. Sorry.
Thank you so much, Erica. And with that, I will pass on to Jen.
Can you see my screen okay? I'll just put it on presenter mode.
So far, it's black to me, but we'll see. Okay, now I can see your presentation perfectly. All good.
Thanks, Gilda. Yeah, hi everyone. I'm Jen Freer. I'm an ecologist at the British Antarctic Survey. And together with Matt Pinkerton in New Zealand, we are heading up the Biogeography Modelling and Management Working Group. So there's 29 members in our team so far from 10 different countries and a really nice mix of backgrounds, expertise, and career stages, which makes it a really nice group to be part of. And yeah, just a heads up and thanks to Matt for all his support as co-lead, sort of as I sort of come into this role, helping to develop this team. So why BMM or Biogeography Modelling and Management? Well, it might be obvious, but these themes have a lot in common. So biogeography is key to understanding ecology and biodiversity and climate impacts. And modelling is a really important part of that for trying to overcome spatial and temporal biases that we have in our observations. And a classic example is modelling distributions of fishes in the Southern Ocean or elsewhere. And models are so much more than that, though. They are essential tools for representing, exploring, and anticipating changes in systems. And this can be biogeographic, but they can also be food web models, process-based models, or even ocean-wide Earth system models. And lots of these models can be important in the context of management. Precisely because they can explore and anticipate change, that advice and evidence can go into decision makers when they try to achieve their management and conservation goals. And just one example might be looking at the overlap between the krill fishery and predator consumption of krill, such as ice fish. But these groups don't work in their own. We are working really a lot with different working groups of SCARFISH to get the voice out and have shared interests. So just some examples. With the data working group, we have shared interests in observations and improving the accessibility and transparency of our fish observations. And with the biology and life history working group, it's been really nice to know what their research interests are to make sure that they align with modelling goals and vice versa. And it's also nice to be involved in the Horizon Scan working group to make sure that, yeah, we are voicing emerging issues and capabilities from a modelling point of view. And of course, it's also great to be part of the outreach ideas and developments to improve diversity and awareness of what we are trying to achieve. So in the beginning, we wanted to have a core activity that would bring everyone together and help us focus our goals. So we decided to develop a peer-reviewed paper, which at the moment is titled Southern Ocean Fish Modelling, a community-led review of approaches, tools, and future directions. And the aims are quite diverse, but we really want to showcase the range of modelling tools available to the community and to highlight resources and databases, and really to make models more accessible to non-modelers and end users. So yeah, This would also involve encouraging best practices and highlighting special considerations when modeling Southern Ocean fishes compared to elsewhere. And we also want to synthesize all of this to look at future priorities and modeling gaps. And really, it's just a way of acknowledging that the modeling landscape is quite complex and can be difficult to navigate if you are new to modeling or are not a non-modeler, but you will use model outputs. So other examples of these sorts of reviews exist. There's an example here from one in Nature, Ecology and Evolution, and it's been a real success. So we hope to draw on those examples and hopefully create a useful resource. So at the moment, this paper is well underway. We have 34 authors and 13 different model types being represented so far. So it's quite chunky and ambitious, but it's really nice to have such buy-in from the community and have a really comprehensive paper. So we have everything from single species models, such as the species distribution models or genetic stock assessment models, and then everything from multi-species Bayesian models to full ecosystem size-based or process-based models. So this is very much in prep. The sub-teams have started writing. We've had several meetings. Yeah, we hope to be well underway by this summer. So that's where we are with that at the moment. But there's lots of other outputs that we are interested in developing as we continue. So just some examples here are some collaborative science papers that will come together over time, providing web resources and just really becoming an active network of BMM researchers. We will be helping to co-convene the fish session at SCAR Open Science Conference. We'll work with the other working groups to produce some data. So hopefully publishing some data from the grey literature and promoting and sharing model outputs such as fish habitat, shapefiles, distribution model projections, food web outputs, etc. And we really hope that they can feed into CAMLR as well as advocating to CAMLR our data needs modeling advances. So that's all ideas and things to take forward in the future. So that's everything from us. I don't have a QR code either, but do get in touch. Feel free to email myself or Matt. And this can be anything if you want to become a member of the group, be involved in the paper that we're writing, or if you have any other suggestions and ideas for us to take forward. And with that, thank you.
Thank you so much, Jen. And I just put in the chat the name of the session at SCAR that you were referring to in case folks have abstracts that they might want to submit for the deadline at the end of the month. And with that, I will pass on to our last working group presenter, Thomas.
Hi, do you see? No, wait, I have to click share. Is it coming now?
Yes, perfectly. Thank you.
Yes. Yeah, so let's finish by the genomics, physiology, and pathology group. So quite a diverse theme in a way where I co-lead it with Julie York and Jin Yoon Kim. And we have quite a large group of people who have been joining us as well. So we've worked on, like you see, like genomics, physiology, and pathology. That's quite a diverse theme. So we've worked on different little things. And one of them is genomics, where we actually more like still brainstorming what we can do because the first genome came out in 2014. And since then, based on NCBI, there's been quite a few more species coming up, mostly orthothenioids and liparids. And that represents a total of 60 or so species for maybe around 250 species, so about a quarter of the species. And with the new technologies now, we get more and more high quality genomes. With so far, there's 12 species that are at the chromosome level scale and 5 that are close to that. But that also means that a lot of them are not at that level and are not meeting the VGP, the Vertebrate Genome Project standards. And as you can see, the taxonomic representation is clearly uneven as well. So we are thinking about maybe providing actions or recommendations on potentially taxonomic priorities for genome sequences, assembly guidelines, whether provide like clear, this is how the VGP is standardizing things, but also reporting the sex, because that might seem obvious, but actually in many cases, we don't know the sex of the specimen that has been sequenced. And trying to name the chromosomes in a uniform way whenever it's possible, especially for example for nothothenioids. But also recommend, and that goes back to like also the data aspect, maybe to try to have the physical sample available somewhere as a voucher in a collection, or deposit some extra DNA extract somewhere. Uh, Report some metadata clearly for like RAD and, for example, nowadays like low coverage whole genome sequencing data, so that they can be more reused by others. Some guidelines for transcriptomic analysis geared towards physiological experiments. So it's really still a brainstorming stage. So if you have any ideas, suggestions, or interested in contributing, like contact us and help us like kickstart this. Otherwise, on the second term, the physiology, we actually have made some good progress on meta-analysis. And that was led by, and is led by Julia York, who is attending right now, about the physiological response of Antarctic fish to heat stress. And so a few of us have participated so far, and we went through a lot of articles and reading abstracts of almost 1,000 papers total, mostly coming from the Web of Science, but also some coming from the great literature of Kamala. And Julia introduced us to Rayan, a tool that allows us to pretty quickly go through the abstract and basically associate keywords to articles. And for us to say, yes, that article is meaningful for the meta-analysis, or no, we can discard it. And out of those 992 papers, we ended up keeping about 30% of them, 35% of them. Most of them have been excluded because they were not experimental, so not really matching the meta-analysis. From there, right now, we are dispatching now the reading of all those articles, like more detailed reading, to try to see if on specific themes, we can actually combine those experiments and come up with a more global understanding. So we see that, for example, nothothenioids and temperatures are a keyword that come up a lot, but eel pound is the second group of fish, and with only 22 articles, so it might be complicated to work on those. And if you're interested in contributing, please contact Julia, because we are still looking for people to help us like go through all those articles now in more detail. Another thing that we've done, our co-lead, Jin-Yoon Kim, asked a few of us if we could help him and his lab like finalize a recent review that was submitted to the special issue of Journal of Fish Biology earlier, well, November or something like that, about the adaptation to extreme environments and a review of Antarctic fish physiology. And last letter of the group is the pathology side, which has the goal to try to evaluate the current knowledge on Antarctic fish pathologies and identify the knowledge gaps, knowing that very few people actually are really focusing on Antarctic fish pathologies. So the actions that we've done so far is that I'm now a member of the Antarctic Wildlife and Health Network, and I represent the fish people to that. And we actually have a meeting this afternoon as well. I also interacted with the SCAR spec group, so the plastic in the Antarctic environment. And if any of you have any interest in like plastic and fish, please join them, because actually I was the only one fish related in that group. So, and I can't join every single group. So there's room for fish people there. And then we are still in the process of assembling a team of fish parasitologists, virologists, health experts for trying to put together a literature review. And the same as for the other aspects, if you want to contribute, please contact us. And the last plug is that we are also organizing a session at the SCAR meeting coming up, this one on Antarctic fish evolution. And so it's the three of us and our colleague Michael Maschiner from Germany. And it's really about fish evolution. We are interested in any group, how they evolve, how they compare to one another, whether it's taxonomy, systematic morphology, physiology, molecular evolution of specific gene family. So just if you have something about that, please come and share your Antarctic fish evolution study with us. And on that, please contact us if you're interested in contributing.
Thank you, Thomas. With that, I will hand off to our last presenter. So that was now the end of our working group presentations, and now my group co-lead Cassandra will talk to us about all of the CAMLR connections and updates and all related work with SCARFISH. So right now, Cassandra, we're seeing your presenter view.
We'll try again. Okay. What do you see now?
Now we see the good version. Thank you.
Awesome. I have to just say it's so amazing to hear from all the groups and all the incredible work that SCARFISH is doing collectively. Just when we all came together to make this group, I think this was the vision for it, and the community was so ready. So we're excited to have this webinar to grow the community and to bring you all in on this amazing work that's happening. So I'm here to talk to you a bit about how we engage with CAMLR, which is the Commission for the Conservation of Antarctic Marine Living Resources, and the work we're doing in that space. And I certainly invite others who work in that space to chime in at any time. Erica, Christopher, in particular, if you have additions to anything I have to say, I definitely welcome those. And just to share that, in addition to being involved with SCARFISH with Gilda, co-leading this group. I'm also Chief Officer of SCAR's Standing Committee on the Antarctic Treaty System, which means that I'm the we're the main group within SCAR that actually brings forward research into both CAMLR and the Antarctic Treaty space. So perfect opportunity to actually make sure that the great work that this group is doing gets channeled into the policy space. So I think most of you are familiar with CAMLR, but they are their office is actually based in Hobart, Tasmania, and that's where their meetings are held. And they have 27 different member states depicted by the flags in front of you, and they make all their decisions based on consensus. And just to share that CAMLR is often celebrated actually as a model of marine conservation on an international scale, because they have this really amazing mandate where they have to, they allow for fishing, but it has to be precautionary, it has to be grounded in science, and it has to take an ecosystem approach. However, of course, there's always limited capacity, and this was part of the motivation for SCARFISH, as Gilda shared, was to really connect that wider fish community, Antarctic fish community, with the CAMLR community. And just to share too that there are two main commercial fisheries in CAMLR, and that's for Antarctic krill, which of course is the key prey species in the Southern Ocean food web. And this is by far the largest fishery. And one of the concerns, which I'll get into very shortly, is that krill is done via these trawls. And when they trawl for krill, they actually catch a lot of larval fish. And there's just a lot of unknowns there in terms of how many they're catching, the species they're catching, the life history of the fish they're catching. So that's one of the things I'll get into more in a minute that SCARFISH is helping with. And then the other main fishery is for toothfishes, which are by far the largest fish species in the Southern Ocean. And they're sold, at least in the United States, as Chilean sea bass. And they're not just an important predator species, but an important prey species as well. And there's also limited fishing for mackerel ice fish as well. So I just want to share a tiny bit about this process, because key to how SCARFISH works within CAMLR is making sure that we actually bring the science in at the right point. So if you guys haven't seen this graphic, it just tries to explain how CAMLR works. And I shared that CAMLAR is really grounded in science. And part of that is this amazing network of science that actually feeds into decision making. So if we're starting at the bottom, there are expert groups and workshops and intersessional groups that work throughout the year, and that feeds into these formal working groups that CAMLAR has. And the main one that we feed into is WGFSA, which just means Working Group on Fish Stock Assessment. And as that sounds, it's a group that actually really focuses on the science that underpins the stock assessments, but also really discusses general fish ecology and life history, especially for toothfish and icefish, but also krill. And those working groups then feed directly into CAMLAR Scientific Committee, which is its main science advisory body, which feeds into the commission, which is the government side, the diplomat side, that ultimately makes the conservation measures that set the policy decisions. And so that would be our total allowable catches and any other rules around fishing in the Southern Ocean. So we really feed in at that fish stock assessment level, and that's through submitting papers and having representatives in the room that can speak to the work. And then we also feed in through the scientific committee, whereas SCAR, I can sit in the room and we can submit papers actually on behalf of the group and the work we're doing. Christopher, is there anything else you wanted to say about FSA before I move on?
Yeah, I mean, I guess the other thing, FSA does have a bycatch component to it as well, where I think SCARFISH could really benefit. And as a matter of fact, one of the priorities of the scientific committee is to start formulating and bringing forward biological parameters for those bycatch species. And bycatch is definitely something that happens in all the fisheries. Specifically, I think if you're talking about smaller species than the krill fishery, because there are actually quite a lot of bycatch species of all different trophic levels in that krill fishery. So that's one thing that we have been trying to reach out and form a more sort of established link with SCARFISH and FSA as well as the scientific committee. So just noting that.
Thanks, Christopher. And I'll move on to this great table, which after I talk, Christopher, you're welcome to chime in too, since you helped create it. But we first brought SCARFISH to this fish stock assessment working group in 2024, and Christopher worked within that meeting too to work with the community and really figure out what are the overarching themes of overlap between SCARFISH and the CAMLAR working groups. Where can SCARFISH really feed in? And if you look at these topics, you'll see that this really cuts across so many of the things you already heard about within all of our working groups. And as Christopher elaborated on, life history traits specific to the bycatch species is really a priority and something that SCARFISH can work on, and including that larval fish bycatch, but other bycatch as well. And so that's certainly one of the priorities of SCARFISH right now, and also this really key area of overlap between CAMLAR and SCARFISH. There's also community ecology, and that's especially diet and ice escapes. And there is a lot of work ongoing in the data group and other groups that's pulling that information together. On connectivity, a lot of the SCARFISH community does work there, as well as on climate change. And climate change really cross-cuts all of our groups. We all touch upon it in some way. And so that's certainly also a priority. Core habitats, that's something certainly that the modeling group and others will be touching on. Plastics is a key issue, and then communication. And this is something that SCARFISH really can tap into, because we have scientists across so many different communities, including other SCAR groups, including SEUS, which is the Southern Ocean Observing System, as well as IUCN, which you heard about, and other research fisheries management organizations. And just to share, you already heard from Becky about how SCARFISH is helping with this new review of Southern Ocean fish species, but this is a really cool effort that I just want to elaborate on for a minute. And Becky, feel free to jump in if I miss anything. But there will be a thorough assessment of Southern Ocean fish species that the IUCN is leading on, and really trying to assess the status of Southern Ocean fish according to the IUCN Red List criteria. These criteria are seen as authoritative on understanding threats to species, like they're actually threat of extinction. And so SCARFISH is helping to make sure data informs that process. It can be hard for scientists outside of CAMLR to know how to access CAMLR data, so we'll be helping with that, facilitating that interaction, as well as compiling data and helping them have access to it and being involved in that workshop. So that's a really cool collaboration between the IUCN, SCARFISH, and CAMLR. And again, just climate change is a big piece of that. Christopher, is there anything else you wanted to say about this table?
Yeah, maybe a couple things. First of all, that IUCN review is just in a few weeks. I think it's mid-March, so that's coming up super soon. So I don't know if there's a possibility of SCARFISH to get anything together to contribute to that. To be honest, I'm not even sure how that review is going to work. And I know there's probably a few people in this group that are going to it, but I haven't really been provided with any information as to what the format or what the inputs or how we're evaluating or what so that's kind of a big weird space. The other thing I want to mention about the data part, and you had mentioned, well, I think earlier when we were talking about data, is commercial confidentiality of CAMLR data. And that's a real that's an issue for a lot of things. However, I think that it depends on what data you're looking at. Certainly, there's a lot of data that's available, and more information is more confidential than not. For example, specific locations of where the fishery catches their target species is the most of the confidential. However, there's a lot of different data available. There's a lot of bycatch information that's available, and there's a lot of different levels to that confidentiality. But one of the things you could do is try, and so certainly I'm happy to work to make those data requests through CAMLR for anything that's available in relation to any species that's happens to be caught, targeted, or bycaught. So yeah, I just wanted to mention those things. The other thing I wanted to mention is the upcoming fish stock assessment working group and scientific committee, and whether or not us as SCARFISH would like to put together something for that to present in the working group. And there will be a couple of us coming to that working group, and I'd be happy to table it or have it tabled through something else to get it on the radar, because that's where really most of the discussion will take place. When it goes to the scientific committee, it's presented and you may get a question or two, but people don't really dig into it at the scientific committee. So really the working group is where we would need to make our impact. So I'll just leave it at that. But anyway, yes, it would be nice to know if we're going to table something. I hope we will. Yeah, thanks.
Yeah, I imagine we will definitely table something. I'm going to give it to Becky, who has been very involved in the IUCN connections and discussions. So Becky, did you want to elaborate on that?
Yes. Well, I was first of all going to thank Christopher just for raising the thing about CAMLR as well. I think one of the things the data group is very cognizant of is that the data is, once you've requested, available. Just a lot of people don't know how to do that. So that's one of the things we will be working on, trying to make that a little bit more transparent and less scary, because a lot of people just go, oh God, what do I do? So, and it's hard to know what data CAMLR has because it is behind all of these walls. And unless you know people, it is very difficult to gauge even the rough metadata of, what decades are we talking? That kind of stuff. So, but it is a very good point that there is a lot of data and not everything has the same level of confidentiality. So there is a breadth. Regarding the IUCN, so I have spoken with Christy, who is the organizer of the workshop. So the way I understand it is that the workshop is just one of the steps. So the assessment of the conservation status of the fish species will carry out throughout the whole year. So the aim is to have it done by the end of this year. The workshop is only one of the core components. I have, however, gotten several requests from people who, similarly to you, didn't know what to expect. So Gilda's posted it in the chat. Beat me to it. Thank you. We are running 2 sessions where I've asked Christy to attend and ahead of it to try to provide some more info about what you are actually expected to bring as experts. As far as I understand, they're still narrowing that down at the moment, but it should give you a better idea of what you should be, what data they might appreciate you having access to or being able to provide, that kind of stuff. So during the 2 sessions will be the same. It's just 2 opportunities for people in different time zones. So if you are able to join one of them, they are open to the entire SCARFISH community. So even if you just have an interest and you want to know what's going on, feel free to attend. And hopefully we will be able to answer some of the questions there. But yes, it is a larger effort across the entire year.
Thanks, Becky, and thanks for liaising on that effort because it's going to be significant. So just to close out, another thing I just wanted to mention that is really exciting for our collaboration within CAMLR and beyond is that we are looking to collaborate closer with the SCAR krill expert group. In fact, we very much got inspired to create a SCAR fish group based on the success of the SCAR krill group that's done a really amazing job of creating community in the krill Antarctic community and actually bringing that into CAMLR as well. So we're actually having a joint workshop with the SCAR krill expert group at the Open Science meeting in August. So for those of you who might be there, please join that because you'll get to actually learn about both the krill and the fish group and help us discuss and prioritize where we can work together. With that, I think we're going to questions and discussion. There's that QR code again for folks who want to join, and I'll hand it back over to you, Gilda, unless folks have other things to add to the CAMLR section.
Thank you, Cassandra. And I just put in the chat the workshop that's going to be taking place between SCEG and SCARFISH at the Open Science Conference. And I just also wanted to emphasize what Becky mentioned about the IUCN preparatory meetings next week. So she had sent that out through the SCARFISH overall listserv. So if you're on that mailing list, you should have received that info. But again, it was in the chat. And even if you're not planning to participate in the workshop of the IUCN next week, but you're either interested in learning more about it or want to contribute sort of what you want to see happen there, this meeting is really about us trying to coordinate that SCARFISH community to both inform how we go into the workshop, but also to help support the people who are going there so that they're representing not just their own voice, but the voice of the wider community. So I do encourage you, if you're interested, to check out one of those 2 sessions next week. With that, I open the floor to any questions or comments on any of the content that we just presented. And thanking you for your patience. I know we're a few minutes late at this point. Also, if nobody has any questions or comments just yet, we also know it was a lot of information to have at once.
Gilda,
Hi,
I'll
if I
just...
could,
Oh, go ahead,
go
please.
ahead.
Go ahead. No, I was just going to say, really encouraging folks to get involved in the different groups and to learn to be able to be involved more in our day-to-day or week-to-week or month-to-month work. And we're just really excited to have you all here. Go ahead, Christopher.
Right. So I just wanted to get back to something we were talking about in the fieldwork section. And I know we had talked about getting together a form to circulate that has to deal with availability of samples. And I'm not 100% sure, unfortunately, where we are. Is that been made available? And
I think Haley, you can speak to that, right?
yeah, Haley would be
Yeah.
the one to speak to that.
Yeah, so I think we're planning on launching it at SCAR Open Science. And so it'll be made available then, but we're also gonna like push it out in other ways. But I think we're just waiting on a little bit of feedback from some folks, making sure that it's all gonna work hopefully smoothly once it actually gets launched, just doing some like testing and stuff like that.
Okay, thanks for that. And that goes in both for the availability of samples on hand, and that could be tissue or otolith or whatever you might have, as well as things that are coming up. Because there are opportunities coming up. I know in the next field season, I'm planning to collect a lot of things. So I would also be happy to collect for anyone that needs any sort of fish samples. I just want to
Yeah,
make yeah.
I think
So
that's
hopefully
definitely a place
okay.
for sure. Thank you. That's awesome. Amazing.
And I've just put in the chat again, because I know it can sometimes be confusing, and this is sort of the best system we have, and I apologize if it's confusing, but there's sort of two ways that you can keep in touch with Scarfish. The first is just being part of our Scarfish mailing list. Maybe many of you already are. Maybe that's how you found out about this webinar. That just means you'll get sort of updates on what we're doing, but it's a more of a passive involvement. And if you wanted to have a more of an active role, then you should sign up for a working group through the form that's linked there. And whatever working groups you pick, you'll be added to those working group mailing lists, and you'll have the opportunity to get involved in the activities that they mentioned there. So just to differentiate, there's the general Scarfish mailing list that's more just updates, and then there's being involved in the working groups and put on the individual working group lists to get more involved in their activities.
There is a question in a chat,
Oh,
Mandy.
great. That's a good question, Mandy. We have a group involved in behavior. Do fish behave? No, I'm just kidding. Uh, I'm wondering under what hat that would potentially fall in terms of what we do. Any thoughts of where that might fall under what we have going on?
Maybe the life history.
Yeah. If I had to give a home to behavior, I would say the life history group might be. I don't know what you think, Erica.
Mhm.
Yes, I think it's very good. Yes, I agree. Yes, because inside the group there is the ecological rules, and I think it's important to consider the behavior, of course. Thank you for suggestion.
Yeah, so that could be if that's something that you
Yeah.
wanted to like bring into the group. I don't think we have a sort of active community of fish behaviorists that are represented within Scarfish, and I think the biology and life history group could be a good place for that. Christopher.
Yeah, I was just thinking about what that means, behavior also. And there are actually a lot of behavioral aspects that we could examine, like species complexes, like whether or not these fish are thigmotaxis like to be up against things, whether or not the fish are solo, whether there's a lot of different behavioral aspects of Antarctic fish. And that's actually a very interesting topic that not a lot of people have really thought about other than perhaps fishermen, and are they close to the bottom or are they water column or things. So it definitely ties in very closely with life history characteristics, but it is an aspect of that life history which of course can change as from larval to recruitment to whatever as they go through various life stages in ontology. So I think it's really an interesting aspect.
Thanks, Christopher, and thank you, Mandy, for bringing that up. Any other questions or comments that anyone wanted to bring up?
So one of the questions I always get is what is the lifespan of our group, and I think this also relates to is there a behavior group, that kind of stuff. It's very much a growing group, right? I don't know, Gilda, if you want to speak to that briefly, but we're hoping to stick around for a while.
Yeah, thank you, Becky. That's actually a good point. So I think we all have varying levels of familiarity with SCAR groups. So just to be on the same page, we're currently an action group. So that means that we have a 4-year term. And the idea is after 4 years, we either say, okay, we've done everything that we set out to do, we don't need to exist anymore, or we apply to become an expert group. And that's the trajectory that the krill group, SCAG, krill action group, and then now SCAG expert group. And that's what we're hoping to follow along as well. So in many ways, our sort of first, and we started at the end of 2024. So we're starting our almost second year now. We're sort of a year and a half actually in. So it's relatively early. It's the first half of our existence as an action group. And a lot of what we're doing is very much grassroots driven. It was the people that wanted to get involved and see the research and ideas and questions that they wanted to see represented in the group. And so that's how we built our working groups. And the horizon scan is sort of a bit apart from that because that has the aim of really informing the next steps of our SCARFISH group, which will be when we become an expert group, and where do we really want to focus our energies. And probably a lot of what we're already doing is going to be highlighted as priorities, but that horizon scan will really allow us to codify that, quantify that, and maybe also bring up other areas that we're not currently addressing. So yeah, so that's the plan. We are from the end of 2024 to the end of 2028. Hopefully at that time, we'll transition to an expert group. And then expert groups, I believe, have also a 4 to 8 year time span within SCAR. I don't 100% remember, but the plan is to keep going. And to sort of change maybe our format, what our specific working groups are, what our outputs are over time based on, well, what we as a community feel needs to be prioritized. So this is very much we really try to make this a bottom-up effort. And so I hope that helps explain a little bit about our timeline. I think that's a good thing to specify, Becky. And Christopher, you had your hand up before.
I did, and my apologies for not bringing this up a little bit earlier. When Cassandra was giving her presentation about various SCAR Camelar ties in and the working groups of Camelar, there is another working group that's called the Working Group on Acoustic Survey and Analysis Methods, and it's also a formal working group. They have primarily studied krill. However, there has been some work in looking at other components of the pelagic ecosystem, which could include myctophids or could include other layers that should be part of that group. I'm wondering if this group finds it worthwhile to introduce ourselves to that WGASAM, acoustic methods, and to try to see whether or not they could assist in using acoustic methods to also study fish in the water column. And I know this isn't strictly SCARFISH, but it kind of is, because you could describe a lot of aspects of pelagic fish with acoustics. You can't necessarily identify them, but at least there is another source of data that could potentially be tapped into. So I don't know if this is worthwhile to bring to that group's attention or just let them stay with krill and forget fish. I'm just throw that out to the group.
I mean, I think it's an interesting idea. I almost wonder if the question is more to the CAMLAR working group, like if they would want to have input from SCARFISH, or I guess maybe SCARFISH has to come to them maybe with a a list of things that we think we could contribute and then do the engagement in that way.
Yeah, I think that they probably don't even know of our existence. So that's why
Yeah.
we could at least say we're a group. Perhaps we could be useful. Perhaps we could provide some information, or you could provide us with some information, or we could somehow work together towards looking at the finfish component of acoustic surveys.
I think why not? Like we could chat with you about that more, Christopher. Yeah.
Go ahead, Jen.
Yeah, just to add, I think that's a really good idea. And there are some people at the British Antarctic Survey who are in the ASAM group who do a lot on acoustics, but also have backgrounds in myctophid fish. So that could be a good connection if we wanted to take it to them. So happy to help with that.
Yeah, I'm immediately thinking of Tracy Dornan, and I'm sure there's a lot more people, but I think there's that community of acoustic fish people that we might want to connect with and see about, yeah, preparing something for WGASAM. Very cool. Well, are there any last questions or comments? We had a bit less time for the Q&A, but maybe we didn't need as much time as was initially allotted, so it might actually work for us to finish right on the 30 minutes past the hour as we had hoped. Well, I hope I speak for all of the SCARFISH leadership in thanking you all for coming and taking part in this inaugural webinar. And a huge thanks to all of the presenters today. Some of you waking up in the middle of the night, Becky, to take part in this. So huge thank you. Huge apology for the technical difficulties in the beginning. This was the first time I set up a Zoom meeting with a registration, and I did not know it would create a separate link to the one I normally use. So lesson learned if you're ever setting up a Zoom meeting in this fashion. And we will prepare a little summary synthesis of this webinar to share with all of you. I think I've hammered in so many times the ways to sign up to the mailing list and the working groups that you'll be sick of it, so I won't mention it again. Yeah, we will be in touch through our SCARFISH listserv as well as the outreach group communications that they're planning in the coming months, and hopefully maybe see many of you at, well, approximately the IUCN preparatory workshop meetings that Becky is organizing next week. And then maybe later in the year at the Open Science Conference. And don't hesitate to submit to any of those sessions that we highlighted dealing with fish at the Open Science Conference. We'd be really excited to see your work there. So with that, I wish all of you a wonderful rest of your day, evening, maybe going back to sleep, all of that. And we'll speak to you soon. Bye, everyone.
Thank you.
Thanks.